everyone. On behalf of the Rose Lab, we will be presenting on building a gene expression atlas of developing brainstem motor neurons as a toolbox to study rare congenital neurological disorders. Here, we study the ocular motor neurons, which are responsible for eye movement. Currently, much is not known in terms of their unique gene expression. We will employ RNA sequencing to develop a transcriptomic atlas of these motor neurons. In doing so, we aim to provide deeper insights into normal development with motor neurons and aid in diagnosing congenital cranial disinnervation disorders.
Studying gene expression profiles of CN3, CN4, and CN6 motor neurons provides a better understanding for cranial disinnervation disorders like Duane syndrome, where non-affected individuals see normal innervation on the lateral and medial rectus muscles from CN6 and CN3 respectively. Those with Duane syndrome often have wrongful innervations from those cranial nerves onto incorrect muscles.
We collected and analyzed gene expression data across various developmental stages, as illustrated in the timeline from E8.5 to E18.5. By dissecting IL-1 GFP knock-in mice at various embryonic ages, we can better understand the dynamic development of motor neurons. The RNA from these isolated cells is then sequenced to produce a count matrix, providing the essential data for our analysis.
We use single-cell sequencing and spatial analysis to retain individual cell resolution and map each cell's precise location in the tissue. To ensure the accuracy of our data, we use advanced computational tools like SC-Dreamer to adjust for batch effects and other experimental variations. This algorithm enhances our analysis by ensuring that our findings reflect biological realities rather than procedural discrepancies.
We use SE Dreamer to generate both unsupervised and supervised atlases. The unsupervised atlas, built without any cell type label information, integrates data based on intrinsic biological signals that it identifies itself, revealing developmental trajectories and enabling us to form hypotheses about motor neuron development. For the supervised atlas, we provide cell type labels to the model, allowing the model to cluster cells accordingly. This enables precise analysis of each cell type and helps identify genes that are highly expressed in one cell type relative to other cell types.
The final goal is to place these refined cell types into a spatial context to understand how different cell types interact with neighboring cells. Because spatial transcriptomics data is not at single cell resolution, this can lead to muddy and non-differentiable cell types. Using SC-Dreamer, we can integrate the two datasets to perform a deconvolution to find the distinct cell types in the spatial data, which we can then compare to light sheet images to validate the anatomical correspondence. By utilizing cell-to-cell communication methods such as cell chat, we can further investigate how motor neurons and other cells in the brainstem communicate.
In the future, we plan to combine cell rank and optimal transport to map cell fate trajectories during embryonic motor neuron development. As illustrated in the figure, we aim to use MOSCOT to align single-cell data across developmental stages by solving an optimal transport problem between time points. Using cell rank, we intend to analyze these trajectories to identify initial progenitors, terminal states, and key fate branches. We will compute fate probabilities and analyze gene expression over pseudotime to reveal when motor neuron subtypes diverge, insights that are critical for understanding congenital motor neuron disorders.